Hello, this is Ed Chapman, and this video cast is going to be talking about meiosis, the other kind of cell division. Remember, meiosis is not mitosis. It's unfortunate that these two words sound so much alike, but they're very different processes. They both are cell division, but meiosis does not produce what mitosis produces. Remember, mitosis produces copies or clones. And these copies or clones mean they're just like the adult cell that they came from. So as cells go through mitosis to increase in number, all they're doing is a form of reproduction called asexual cell reproduction. And there is no variation being created. So each individual that comes out of mitosis is just like what went into mitosis, the original cell. Meiosis is different. Meiosis is a process that cells go through to make a special kind of cell called a gamete. And the gamete cells are specifically made for sex. All right? Males produce in their testes, if they have testes, not all animals do. You can also have sperm cells produced by plants, in which case they're inside of grains of pollen. But let's focus right here on animals. Gametes in males are called sperm cells. They're made in the testes. And females produce their version of a gamete, which are called egg cells. And gametes are fundamentally different from all other cells in your body. Gametes contain different genetics. Specifically, gametes have half the normal number of chromosomes that you find in body cells. So body cells, otherwise known as somatic cells, this might be a new word for you guys, they're what we say are diploid. They have two copies of each chromosome, or a pair of chromosomes, like we saw when we looked at karyotypes. For example, in human body cells, there are 46 total chromosomes, but they're arranged in 23 pair. And if you think about it, this is because every human receives 23 pair from mom and 23 pair from dad. So you, as a human, are the result of the fusion of two gametes, an egg cell and a sperm cell. And when they each bring their 23 chromosomes to the party, so to speak, you end up with 46 total chromosomes. Now, gametes, as I said earlier, are haploid. These are otherwise known as sex cells, and they contain half the normal number of chromosomes because they have to. Two gametes are going to combine to produce a new offspring. So gametes have to be haploid. This word means half the normal number of chromosomes. And for example, in human sperm cells and human egg cells, there are only 23 chromosomes. They no longer are paired up. Now, we can illustrate this with these little easy to follow pictures here. If we have two haploid gametes, over here on the left, that represents an egg cell with its 23 chromosomes. And over here on the right, we have a sperm cell with its 23 chromosomes. When they combine, you end up making a zygote cell with the total 46 number for chromosomes. So this zygote is what we call a 2N cell, and it has pairs now for every one of its chromosomes, and it's the normal number for humans. Now, after fertilization, the zygote is going to go through a whole bunch of mitosis, and this is going to lead to a lot more cells. At first, these cells are all identical, they all look the same, but at some point they're going to start to change. But while they're all the same, they have the ability to become any other kind of cell in the body. They could be nerve cells, muscle cells, brain cells. So these cells that have this ability to develop or differentiate into any other kind of cell are referred to as stem cells. And they're very important to biological research and genetics. Now, the zygote goes through mitosis, as I said, to grow into an embryo. So you start out with a single zygote cell. In case of a human, it's 46 chromosomes. It's going to go through mitosis and divide into two cells, each with 46 chromosomes. And it's going to continue to go through mitosis. And here we have 4 cells, each with 46 chromosomes. You know you're looking at a mitosis process when the division produces identical daughter cells. So in each of these cases, we're not changing chromosome number. The number's staying exactly the same. So zygotes go through mitosis to basically grow. And so after a whole lot more mitosis, you end up with what might become, in this case, a recognizable human fetus or human embryo. Now, do you see how meiosis is necessary to reduce the chromosome count of gamete cells by half? When egg and sperm cells combine, it restores the full number of chromosomes. That's a really important idea. If cells in testes and ovaries did not go through meiosis, then when they produced gamete cells, you'd end up with a 46 chromosome sperm cell and a 46 chromosome egg cell. And when they fused during fertilization, you'd end up with 92 chromosomes, which is not a normal number, and that wouldn't be survivable. That wouldn't work in the case of humans. So I'm hoping you see this point. Now, altogether, let's try to summarize it. If you start out with a body cell, okay, that body cell can do one of two things. One thing it can do is it can go through mitosis or mitotic cell division and produce more body cells. In this case, all the cells are going to be the same that it makes. Or this body cell could go through meiosis and it's going to make sex cells, otherwise known as gametes. Now, if the body cell is like skin or liver or bone or nerve, those cells only go through mitosis. But in the testes of males and the ovaries of females, some of those cells have a special ability to do a different kind of cell division called meiosis to make what we call sex cells. Now, remember, body cells are diploid. If you take a diploid cell through mitosis, you end up with more diploid cells. But if you take a diploid cell through meiosis, you end up with haploid cells. And we have a symbolic system for this. We refer to diploid as 2N and haploid as a single N. Kind of like a little algebra here. So 2N means 2 copies of each chromosome, and N means only 1 copy of each chromosome. Kind of an easier way to notate this rather than writing out the words diploid and haploid. All right, now I mentioned earlier that when zygotes go through cell divisions, many mitotic cell divisions, they become stem cells, which is what you see right here. But at some point, these cells start to go through a process called differentiation, or they start to take on different characteristics because different genes are being activated, switched on or switched off in these cells compared to their neighbors. So they follow a different developmental pathway. So in this case, we have the fertilized egg or the zygote of a starfish that obviously is going to grow and develop and differentiate into an adult starfish. Down here, same thing happens in plants. Here we have the fertilized egg of a plant cell that's going to go through a similar process to produce an adult plant cell with all of its differentiated tissues and organs like roots and leaves and stems and mesophyll and epidermis and all the things that we've mentioned in other classes. So the process of differentiation is important because it turns a stem cell into different types of cells. Okay, and the process of differentiation is controlled or regulated by the switching on and switching off of different genes inside of each cell as it differentiates. Pretty cool stuff. All right, so now let's look at how meiosis actually happens. And if you remember when we studied mitosis, we learned about prophase, metaphase, anaphase, telophase, and then interphase, and then maybe prophase, metaphase, anaphase, telophase, cytokinesis again, and around the cell cycle goes. Meiosis has something similar, but it's a little bit different. So we're going to be eventually comparing and contrasting meiosis and mitosis. But before we can get there, we have to focus a little bit on how meiosis itself happens. Now, in meiosis, cells actually go through 2 cell divisions. So that means you start with 1 cell, you get 2 cells, and then you go another division and you get 4 cells. So meiosis is over when you have 4 cells. And remember, meiosis makes gametes. So you could produce 4 egg cells or 4 sperm cells. Now, meiosis 1 is the first division, and the most important thing that happens in meiosis 1 is that the pairs of chromosomes are pulled away from each other during anaphase. In meiosis 2, the most important thing happens is that the chromatids are separated from each other during anaphase 2. And I'm going to show you this on a diagram, so don't worry about trying to visualize this just yet. But 2 most important things to remember about meiosis is meiosis 1 is separating pairs, and meiosis 2, the second division, is separating chromatids. All right, now in this picture, we have a cell right here going through meiosis. And notice in this interphase cell here, the chromosomes are uncondensed. So to get from here to here, the chromosomes have condensed. So this is a prophase cell. But notice something really strange is happening here. Okay, this is interphase. In here, we did not see this in mitosis. In meiosis 1, the first part of meiosis, the first division, in prophase 1, the chromosomes pair up with their homologs. So this is what we call a homologous pair of chromosomes. So in this case, we have this reddish one and this orangish one pairing up and getting very close together. And the same thing's happening with these 2 smaller bluish colored ones. The dark blue one is getting very close to its homologous pair this light blue one. They get very, very close. So that's what happens in prophase 1. And then it's pretty much like we saw in mitosis. Metaphase 1, they line up at the equator or the metaphase plate of the cell, being pushed and pulled there by the spindle fibers. In anaphase 1, the spindle fibers pull the pairs apart. Remember, it's separating the pairs of chromosomes, not the chromatids. They're still stuck together. And then finally, we have telophase 1 and cytokinesis, where this is going to divide into two completely separate cells. All right, so now let's go down here to the second division cycle. Notice now we're starting with two cells. In each of the two cells, this one up here on the top and this one down here at the bottom, they each only have 2 chromosomes. We started out up here with 4, a 2N cell, and down here we've now made 2 haploid cells. They only have 1 copy of each chromosome. And meiosis 2 is just like mitosis. We're going to line them up during during metaphase 2. They're going to line up in the middle in each cell. Spindle fibers form, push them to the middle. Anaphase 2, sister chromatids are going to pull apart. Telophase and cytokinesis, we end up with 2 sets of 2 cells, or 4 total cells. And these cells here could go on to become egg cells or sperm cells, depending on which sex animal or plant they're taking place in. All right? Now, you may have noticed that there's something going on up here that's different. That's the pairing. So please notice that the homologous pairs form during prophase 1. That's important. And you may also have noticed that there's this weird overlapping here. It looks like the tan one and the red one are switching tips, and that's exactly what's happening. So during, go back here. So during prophase 1, the tips of the homologous pairs of chromosomes switch places. Okay, so what you end up with here is a chromosome here that is a mix. It's a combination of the two. So the sister chromatids are no longer identical in this tan one here, and the same thing for the darker red one. The chromatids next to each other are no longer exactly the same. It's kind of a little bit easier to see here. So this chromatid here on the bottom's got a little bit of tan on it, and this chromatid here at the top has a little bit of red on it. So they have traded genetic material, all right? And that happens during prophase 1. All right, so this weirdness that's happening during prophase 1 is called crossing over. So if we look here, this is a little easier to see. When the homologous pairs form in prophase 1, they get very close to each other, and the tips actually break off and switch places. So you end up shuffling or mixing the genetic material that's being carried on these chromosomes, which introduces some variation. So they trade tips, and this produces chromosomes where the chromatids are no longer identical. Okay, as I said, this process is called crossing over, which kind of makes sense. That's what happens. And it shuffles or mixes the genes, so at the end of meiosis, you can end up with gametes that are not identical to each other. They contain different combinations of genes from the mother and the father. Okay, so this increases something called genetic diversity, which makes it so that every baby that's born, every offspring that's produced, no matter what you are, humans, plants, dogs, cats, starfish, if it's sexual reproduction and involves meiosis, you're going to end up with offspring that look different from each other. It's not producing clones. So cloning can be really good. Cloning is an excellent way to copy yourself. It's fast. Organisms that reproduce by cloning are reproducing asexually. They make copies of themselves. In this picture here, this is a sea anemone, and what's going on here is the sea anemone has reached a large size. It's very well fed. It's getting lots of food, and it will literally split itself in half and clone itself. So you'll end up with 2 sea anemones right next to each other that are exactly the same. So cloning means you can reproduce faster, all by yourself. You don't have to find a mate. All your clones are copies of you, and they're just as healthy, just as genetically fit as you are. And this is going to work really well if the environment you're living in is nice and stable and there's lots of food and it doesn't change a whole lot. All right. Now, diversity is better, and meiosis allows for the production of diversity because it produces gametes, which are involved in sexual reproduction. So females make many different many genetically different egg cells, and males make many genetically different sperm cells, and then you combine one egg and one sperm. That happens randomly. So this is why it's impossible for two parents to produce the exact same offspring twice, because the odds of two exactly the same sperm cells and two exactly the same egg cells finding each other between the same couple is astronomically weighted against that happening. All right, so what happened here? Hold on just a second. All right, so populations produced by sexual reproduction contain many, many genetically diverse individuals. So if things get bad or the environmental changes or it's like, for example, a disease hits, there's an odds that some of the offspring are going to survive. If everybody's a clone, like we saw with the sea anemones, if you just copy yourself, everybody has an equal chance of dying from the disease. So it's likely the whole population is going to be wiped out. So if you imagine a population of humans where everybody is a clone, I found this on the internet. I don't know who this guy is, but if you imagine a disease hits and everybody's genetically identical, so that means everybody's just as susceptible to disease as the person next to you, the good chance is everybody's going to die. So imagine everybody dies. So that population goes extinct. But on the other hand, if there's lots of diversity and everybody's different, slightly, genetically different, then if a disease hits, the odds are that only some people are going to be wiped out by the disease and there will be survivors. So that's always better. Okay? So genetic diversity helps species avoid extinction. And this is why sexual reproduction has become so popular among multicellular land organisms. Things on land experience more changes more often than things in the water. So if you look at nature, there are lots of animals that reproduce asexually in the water, like sea anemones and jellyfish and starfish and things like that. But if you come onto land, that reproductive technique is a little less common because water temperature pretty much stays the same, especially if you're in deep water, but air temperature and rainfall and climate and all those things are much more changeable on land. So genetic diversity produced by sexual reproduction has become the way large animals and plants reproduce on land. Now, comparing these side by side, remember I told you our goal was to be able to compare and contrast these. Over here on the left, we have mitosis, and in mitosis, you just take a cell through a cycle that replicates. So you end up with daughter cells that are 2N, just like the original cell. So in this cell here, we see 4 chromosomes, and down here at the end of mitosis, you see 2 cells, each with 4 chromosomes. Now look over here at meiosis. Same original cell, 4 chromosomes. Each chromosome duplicates itself during the S part of the cell cycle, but we go through one cell division that separates the pairs, so we end up with 2 cells with only 2 chromosomes each. So now these cells are officially haploid. And then the second cell division, the second round of meiosis, meiosis 2, produces 2 cells from this one cell, and we get that by separating the sister chromatids. All right. So at the end of meiosis, you end up with 4 gametes, which could be egg cells or sperm cells. Over here, you only end up with 2 cells. So I'm hoping by having them side by side here at the end that you can see what I've been trying to talk about here. Thanks for listening, and we will stop there.